Good morning. My name is Janie Montblanc. With me is our co-coordinator, Corey Guker. And on behalf of the Great Basin Fire Science Exchange and our partners, I would like to welcome you to the first of 4 webinars in our series on seed innovations for Great Basin landscapes. Today's webinar is on seed zones and adaptive traits. So I will stop sharing. You can, Corey's going to put in the chat the link for how to get to this page if you want to register for other webinars in this series. So I'll stop sharing that and I'll share today's talk. All right. We have 2 speakers that will each present for 20 minutes, and then we'll have 15 minutes for questions and discussion. The first talk is Setting the Landscape for Native Seed Technologies, Balancing the Need for Wild Adapted Native Seeds with Modifications Needed for Agricultural Production and Large-Scale Seeding, presented by Beth Leger from the University of Nevada, Reno. The second talk is Guiding Restoration with Seed Transfer Zones, Concepts and Applications, presented by Rob Massatti with the US Geological Survey. Before I introduce our presenters, I will go over some webinar details. We're using Zoom Webinar as our platform, and its only drawback compared to meeting is that attendees can't see other attendees. So if you would like, you could enter your name and affiliation in the chat for a better sense of community on the webinar. But before you do that, please make sure you go to the 2 drop-down arrow in your chat and change the audience of your message from panelists to panelists and everyone. We will also be using the chat for questions, so please type in your questions at any time, and myself or Corey will field them to the speakers after the presentations. Now I'll welcome our presenters. Beth Leger is a foundation professor and the director of the Museum of Natural History at the University of Nevada, Reno. She studies plant population biology and works on local adaptation and rapid evolution of native and invasive species. She also gathers basic ecological data about rare species, natural communities, and invasive species control, providing information that's necessary to make appropriate management and restoration decisions. Rob Massatti is a research ecologist with the USGS Southwest Biological Science Center in Flagstaff. He studies how species and their genetic variation are distributed across Western North America and investigates the historical and contemporary factors influencing these patterns using genetic, environmental, and distributional data. He uses this research framework to place findings in the context of restoration and native plant materials development. All right, welcome Beth and Rob, and thanks for presenting today. I'm going to stop sharing Beth, and you can take it away.
Okay. Can you see everything?
Yep.
Okay. Nice to see you all virtually. I can see that there's almost 80 people here, if you can't see that, and lots of friendly names. And I wish I could see your faces, but it's nice to see your names. Thank you for joining this webinar series. This is possibly the longest title I've ever had on a talk, but Janie just read it for you. I wanted to highlight the other talks in this series. Today's theme is natural selection and adaptive evolution. And then the upcoming talks are about modifying individual seeds for particular purposes, and then modifying seed delivery. And I'm going to talk a little bit about sort of how evolutionary history can guide choices that we make here. I'm also going to talk a little bit about agricultural production, which we don't have a group talking about this, but I think it's a huge area that's ripe for technology and innovation. And then we're going to end with a really fun discussion of what managers are thinking about these different issues that Mark shared with me, and I'm super excited about it, and a little chat about where we are with that. So that's the stage. I guess the bigger stage actually are these amazing plant communities where many of us live. So I always like to take the biggest, broadest picture possible. And when you step back and you remember that these plants have been in this landscape for a very, very long time, way longer than humans, right? So we are a blip in their history. Some of the estimates of how long particular plants like sagebrush have been in North America they're kind of hard to find, but somewhere between 13 million years and 23 million years, which is a huge window, that's when the Artemisia genus evolved in North America. And this genus has been here through tons of change, right? Change that we cannot even imagine. And all of these plants have, right? So they move, they evolve, they innovate, and if they cannot live in these environments, we're not looking at them, right? So we are a blip, and they have this huge evolutionary history where they have evolved all of these adaptations. And so when we approach ecological restoration, I try to do it with some humility, right? And understand that my idea of what makes a successful plant really has to be balanced with what the plant is telling me. The plant is the one, and I'm very anthropomorphic about plants. What I'm saying is, evolutionary speaking, the plants that had adaptations that allowed them to survive, those are the offspring that we're looking at today, right? And if they do not have what it takes to hack it in the system, they are not here. So we have to be a little bit humble when we try to change their traits with technologies or otherwise. That said, there's a lot going on that was not happening, for the last 13 million years. And that's humans lighting things on fire constantly, right? We know that we are a huge disruptive force in these systems. We also know that we bring in a bazillion invasive plants. We've changed the grazing systems. We're warming the climate. So there is a lot that we are doing. It's not like these systems are all ready to become, to go back to being biologically diverse after these kinds of disturbances. There is a place, I think, for humans to sort of nudge these communities back to a natural setting. All of us on this call know that you cannot wild collect seeds at the scale you need to fix this kind of problem. And so we are doing this sort of new experiment called growing these very, very old and ancient plants, right, in an agricultural setting. This is a beautiful Asteraceae forb growing in hedgerow farms in winters with, sort of our technology that we have. So this is more and more common. And myself and some co-authors, so Allison, Owen, Charlie, he's at UC Davis, Todd out of Australia, Christina Hufford in Wyoming, Karin in Utah State, we got together and chatted about the things that might be happening both on purpose and accidentally when we are growing native plants in these agricultural settings. And the reality is, no matter how hard we try, bringing native plants into ag systems or restoration systems in general changes them. It can change them in small ways or in large ways, but it is actually inevitable. And it's here that I think we can use technology to kind of mitigate some of these changes that we might be doing accidentally. And I think it's a huge area ripe for innovation and thought. So native grasses have been in production longer than native forbs because as humans, we're super good at growing grass in rows. It's very cooperative, but we are more and more growing native forbs. And one of the things that my co-authors and myself noticed is that a lot of the methods that we're using to grow these crops are borrowed from food production agriculture or fiber production agriculture. Humans have domesticated crops and we've been growing them very successfully in rows for a long time. And those practices have been the leading edge of how we've been developing native plants. And there's some reasons to think maybe a little bit about that before we just march forward with this. So in conventional agriculture, there's some huge differences than what we need in ecological restoration. And one thing that is obvious is that these environments are really controlled. We can plow them, we can make furrows, we can water them, we can change fertility, we can change planting time. We really have a lot of control over this environment, which is not true when we're doing restoration. Another super key thing is that most crops are annuals and mostly there's one generation per planting. So if you make a mistake and you plant the wrong variety, it doesn't matter. You can just go back in and do it again and do it again and do it again and do it again. That is not the case in restoration, right? Where we're usually planting one generation and we don't have do-overs. In conventional ag, the end use is human-centric, so we know what we want and we can drive the bus that in the one direction or a couple directions. We like uniformity. We want everything to come up at the same time, mature at the same time, harvestable at the same time. And it's not that genetic diversity is not important. We know that genetic diversity, and especially in native food crop relatives, is super important, but we tend to hold that off site. So we'll put a little bit of diversity into a field, but we have the capacity to bring genes back in as we need them. And again, that's a contrast to ecological restoration. And it can get confusing because this step where we have plants in rows, this is where those, uniformity, we need to be able to collect the seeds, we need to, know when they're going to mature. All that stuff is important to us for a very short period of time. But remember, these seeds are going back into the wild. The ultimate goal is very different. I kind of think of it, and here's some, obvious things, there's more. We need to have many generations coming from that single planting. The final growing environment, anyone who's tried to plant a seed in the Great Basin knows, like, dude, we have no control over anything. We rarely have water. The end use, it's not humans driving a single use. Everybody who lives in this system needs a different thing. If you're a plant, you need a nurse plant. If you're an animal, you need food. If you're a pollinator, you need flowers. Everybody needs a different thing. So it's not just like one use driving the boat. Hence, we need variation among plants. And genetic diversity, it has to be there at this step. We have to pull all the diversity that these plants are going to need for the next 13 million years. We have to pull that as much as we can from the ag system, right? We're not going to go back in and keep putting new genes in there. So superficially, it can look really similar to conventional ag, but it's not. And so I kind of think of this as sort of an hourglass shape. So these native plants, they've been there at the top of the hourglass. They've been on these landscapes for millions of years. Then we are this little narrowing. We're like, okay, we need you to grow and be able to plant in a wild system through our ag process. And then the hourglass widens again. We need them to be able to hopefully be here for the next 13 million years. So we are a blip in their time frame. And one of the things, that we have to think about, right, is I said intentionally and unintentionally, this changes things. Obviously, this is a super different growing environment, and plants are being exposed to natural selection in this environment and this environment. And as you can imagine, they're quite different. So some of the things that happen to our native plants when we put them in these systems, it's a sort of domestication syndrome. And again, it can be intentional or unintentional. So in domestic plants versus many native plants, right, you lose seed appendages, you don't have dormancy, you want everybody to germinate rapidly, seed retention, uniform growth and flowering, fast growing, big seeds, reproduce by seeds, make a lot of them. This one's pretty fun, larger, fewer pollen grains. That's not something I had ever really thought about. But when you think about these systems, we're planting these plants conspecific so closely that the pollen is literally falling, from the anther to the stigma. And large pollen is actually more competitive on the stigmatic surface. It can get down and fertilize seeds faster. And so in these systems, you have very rapid selection for fewer, larger pollen grains. Whereas obviously in the wild, right, that's like, you kind of need your pollen to be able to get across these jumps. So that's a fun one. I don't know that anyone's ever looked at it, and it sounds terrible to me looking at pollen under a microscope, but it's one of these things that could change in native plants through agriculture accidentally. Native plants often have huge appendages. They have a ton of dormancy. They stagger their germination. They throw their seeds everywhere. Like, all of these things are challenging for us growing them, but they are apparently adaptive in the wild because they evolve over and over and over again. So I'm going to give you about 4 different examples of some of these sort of domestication traits that we either intentionally or unintentionally sort of foster in our native plants, and some examples of how I think seed technology can help us overcome these issues. And one of them, we're going to start with seed appendages because they're everywhere and they're a pain. Like, I understand. So this is one of my favorite plants. This is Elamus squirrel tail, and it has these huge awns. And if you've ever harvested seeds of squirrel tail, you know that you can take them off the plant and you can put them in an envelope, and it's like a one-way situation. If you ever take them out again, you cannot get these things back in there. These awns are really, really, really hard to work with. And we can't store them like this, like, it's insane. And we can't get them, out in the field with these huge awns on them. So there's often this awn removal step that needs to happen for us to put it through our little narrow bottleneck, right, of restoration. That said, these awns perform a major function for plants. This is an erodium seed, an invasive forb, and it's stuck in a piece of clay. When you wet the awn, it straightens out, and as it dries, it literally drills the seed down to apparently the appropriate depth for this plant, right? So the seed has this own technology itself. I also have a cameo here, I forgot to tell you, from Sir David Attenborough. He's going to tell us a little bit more, 30 seconds of Sir David telling us more about these awns. This is like a super cool adaptation. This is Avena fatua, wild oats. So how great is that, right? So who says plants can't walk? And we know that microsites are important, and plants have evolved ways to find those microsites on their own, right? They literally can walk. When they get there, they can uncurl and they can plant themselves. That is a pain if you're trying to do ecological restoration. And so we have developed this technology. We have technology to remove awns, and then we have technology to put seeds in the ground at particular depths that we want them, right? Some of our speakers are going to be talking about how to overcome this planting barrier and this microsite barrier when you don't have the ability to, drive a tractor, because there are places where you can't do this, or say you don't want to do this level of disturbance. But we're already doing these innovations, helping plants get back to, the places that they need to be once we've removed their own technology. Another kind of cool example. So when we remove awns or we remove seeds from their seed coats, it has other effects besides planting. This is kind of a fun one. This is an example from a really invasive annual grass. This is Aegilops transialis. It's a relative of wheat from Eurasia. You would absolutely never plant this in North America. It's a total weed, but it has this cool seed ecology that Andy Dyer, he was a postdoc in the lab at Davis when I was there through my PhD. And so I'm just bringing this in as an example of how we can change seed behavior by removing them from their structures. So just like wheat, Aegilops has 2 seeds per spikelet, 2 seeds per node. And one of them is huge, like over 2.5 times bigger than the smaller one. And they disperse together, they break up, and in the wild, they just go into the ground together. So 2 seeds together in this little spikelet. And interestingly, when you plant them like that, the big seeds, there's no dormancy, they just germinate. The small seeds, on the other hand, are affected by their neighbor and by being in the seed coat. They have a little bit of a bet hedging strategy with these 2 seeds together. So Andy did this cool experiment where this is the big seed just grows. It's a weed, grows. The small seeds are more stubborn about it. And this is the percent emergence of small seeds that Andy planted still in that spikelet. So if it has its neighbor, its big neighbor, it does not grow like at all. Even if you take that big seed out, it doesn't grow, the little seed. It's just very dormant in there. When you remove it, you start getting some germination of that small seed just from being out of the seed coat. And then if you wet it multiple times, you actually get more germination of this little seed, even when it's in the spikelet. If you take its neighbor away and then wash it, it will germinate, right? So you're sort of changing the dormancy of this plant by adding water. So Andy had this theory that maybe there's some chemical on this on the outside of the seed that's suppressing dormancy when it's in the spikelet. And he did this other little experiment where he washed those spikelets or washed those little seeds out of the spikelet different times. And yes, the more you wash them, the less dormant they get. So that's consistent with sort of this, forced sort of bet hedging strategy in this plant, that half the seeds are ready to germinate and then half of them are sort of waiting for either their sibling to go away or multiple washes before they'll germinate. Sort of a, just in case everything's terrible, you do still have another plant out there. So if I was looking at this, I would say, okay, this plant is telling me that suppressing germination is a good idea if I was going to plant this in a restoration. And I might try to plant the larger, they're going to get separated in the seed cleaning process. I might separate the large seeds and the small seeds I might force some sort of dormancy on those small seeds with some sort of coating or some sort of technology to keep that hedging in there and let those large seeds be non-dormant, because that is what this plant is telling me works, right? So yes, we get it, and we can innovate that. Not all plants are exactly the same. So this is, again, squirrel tail, my favorite. This is a very common plant around here that's very cooperative. And in contrast to Aegilops, it actually likes to germinate quickly, from what I can tell. And so we did an experiment where we planted wild collected seeds, did a precision seeding experiment, planted them in October, and we followed them for 3 years. And what we found, this is a cute little 1-year-old plant. What we found is that they emerged super quickly. This is just a count of the number of seeds out of 1,000. This is one site with Elamis multicetus. Most of the seeds germinated immediately in November. And then there was a sort of a tail, there was a natural tail, but about 90% emerged before February. We tracked survival. Just from this, I would say that early emergence, like the plant is telling me that early emergence is a good idea, but we also track survival. And so these hash lines are seeds that have survived. So we're seeing some survival across the board after the first year. In the second year, we start seeing these early seedlings pull away. They are surviving at a much greater rate. And by the third year, survival was 8% overall, which is actually not terrible. And we're seeing very, very few. This is actually one plant that germinated in February and one in December are still alive. So this is telling me that in this system, I would aim for early emergence, because that seems to be the best way to become a plant. And if I wanted to bet hedge, maybe I would delay germination, but for a very small percentage of the seeds going in, because this tells me that in most years, early germinating seeds turn into plants. Okay, the last example is seed retention versus seed shattering. This is Indian ricegrass. It's named because of its importance to multiple indigenous groups in this area. Areocoma is the new genus. It's a wonderful plant, and it is also kind of a pain. It has indeterminate flowers. The seeds ripen over a long period of time. As the seeds ripen, they fall to the ground. And that tells me that that strategy works to keep this plant on the landscape, that if you stagger your seed production and germination, you're more likely to have your seedlings turn into plants. I think I'm going to skip Sir David. He had something to say about this, but just in the interest of time, I'm going to skip it. But the reality is that whole strategy that works really well for Indian ricegrass in the field is a giant pain when you try to grow it in the farm setting, right? So what you need out here in the wild is plants that shatter and make seeds over a long period of time. What is handy in the ag field is plants that will actually hold their seeds so that you can harvest them. And the way that we sort of used to approach this sort of problems is by domesticating them. So the major cultivars that you can buy of Indian ricegrass, they have been selected because they are more prone to hold their seeds and not disperse them. So we have done this sort of work to go find sort of weird populations that hold their seeds and have less dormancy. And those are the cultivars that we're using now, despite the fact that across the range of the plant, most populations throw out small amounts of dormant seeds continually. And so sort of our previous approach to these challenges was to domesticate and change the plant themselves. But I think where we're at is that we should start innovating ways to harvest plants that are challenging. Don't change their genes, change our relationship to them. So we've grown, this is Terra Decuros. We've grown some of these really challenging, explosively dehiscent plants. We've tried growing them on weed fabric and sweeping them up. This is a great way to collect a lot of rocks, we found. So what we found that works better is if Terra just harvested the plant at a particular time. This is a very high tech, put it in a box, cover it up, and collect the seeds that way. Traditional ag has some approaches. This is Lotus corniculatus. It also has explosively dehiscent pods. And my understanding, when they produce this in an agricultural setting, they actually spray the plant with sort of an adhesive, and it keeps the seeds in the pods, even though they're ripening on the plant, right? And so I don't know anybody doing that for native plants, but it seems like it would be really great for some of these things. Let's not breed out this seed shattering. let's try to, hold these seeds in some way that we can use them, but keep those genes in the pool. So just to wrap up, that was just a few examples. If you look at this list, there's a million things that we want to keep in our plants that agriculture and restoration is trying to sort of push out, not on purpose. It's just a requirement. And so this tension between native plants and what they need and domestication plants, what we need, this is where I think technology fits in. We can try to innovate ways to keep the traits that keep the plants around for 13 million years. At the same time, it lets us grow and plant them in restoration. So again, I think technology can help us through some of these barriers. I think it's important to let the plants tell us what they need before we go innovating. Let's figure out what they're telling us works. Again, innovate, don't eliminate. And it's a huge area for creative people to get really, to experiment and collaborate and get these seeds back into the wild. Okay, and that is my last slide. Thank you all.
Great, thank you so much, Beth. If people have questions, we're going to move to Rob's presentation next, but if you have questions right away, go ahead and put them in the chat so you don't forget them, and we'll come back to them after Rob's talk.
I'm trying to stop sharing. It won't let me. Oh, there we go. I hear it. Oh, I got it. Okay. Great.
Great. Thanks, Beth. All right. Hi, Rob.
Hi. I can use my camera so I can show you all my smiling face and show you how happy I am to be here before I turn my camera off.
Thank you.
Let me see here how I can share the screen. And let's see. Can you see this?
Yep, looks great.
All right. Well, it is really a pleasure to talk to you all today, and especially to follow Beth. Thank you, Beth. That was really fantastic. And I'm going to hide myself. Then, okay, so I really appreciated Beth's evolutionary perspective, and I'm going to be convincing you that we actually need to be incorporating more of that into our native plants. And also, ultimately, what I would like to convince you of is that we'll actually simplify our restoration guidance in terms of the native plant materials that we're using and how we're developing them. So today I'll be talking about concepts and applications of seed transfer zones. Oh, like me. Okay, so I'm going to try to skip through some of the stuff that we all know pretty well quickly so that we can have more time for questions at the end. Everyone here knows that restoration is difficult and that there are site-specific challenges that have to do with the land and the species and disturbance. And there are also a lot of procedural challenges, some of which Beth touched on, including how do we restore, what do we use for restoration, what do we do about changing climates and invasive species. A lot of what I do for Colorado Plateau Native Plant Program drills into the what of restoration. So what are the native plant materials? How do we use those plant materials best? And how do we develop new materials? And that in my job today here is to really tell you about that. And when we think about this what, we really have great guidance available for us in the form of, as exemplified by the National Seed Strategy, which I think many of you are familiar with. The mission here is to ensure the availability of genetically appropriate seed. And I really want to highlight that genetically appropriate portion of this because in some ways it guides what I'm going to talk about in the way we think about restoration into the future, not that it's necessarily how we do everything right now. If you're not going to the seed conference, you should go. Okay. So many of, or there are probably several authors within this webinar today that were authors on this paper here. And really what I'm trying to show you is what evidence supports the use of this genetically appropriate idea. And it's really the idea of local adaptation, which has been really demonstrated for hundreds of years. I think anybody who has experience and a love of growing plants understands that there's a lot of variability in plants and that you can't just grow anything everywhere. This is an example here that is drilling into the Intermountain West in terms of a meta-analysis that looks at the strength of local adaptation across species. And these authors summarize really nicely the signatures of local adaptation in terms of number one, is there among population variation? By and large, overwhelmingly, yes. Are those morphological traits associated with environmental gradients? Overwhelmingly, yes. How often does local outperform non-local? It's a little bit more mixed, but still there's a strong signature that if you use something that we consider genetically appropriate, we can have better restoration outcomes. This also highlights that there's a lot of variability among species, which necessitates ongoing research on our priority restoration species. Okay. Here we go. So when we implement genetically appropriate restoration, what kind of tools do we have? We have provisional seed transfer zones, which hopefully all of you are familiar with. The Bauer zones are on the left, and they're used quite frequently to demonstrate this concept. Very quickly, these are used for species that have no other information available. And if we think about using these, we can apply genetically appropriate restoration techniques to any species to support species diverse restoration mixes. And at the end of the day, for any of you who isn't familiar with this, it's a color matching game in terms of using matching your restoration site to your restoration material within the same colors to improve restoration outcomes. I'm gonna bring up Ammonic level 3 ecoregions several times throughout this talk, so I'm showing you here. And Ammonic level 3 is implicit in both of these figures as a constraint, right? So the idea being, even if we have a restoration site in this pink in the Southern Colorado Plateau, it might not be the best idea to pull a native plant material from further to the northwest, several ecoregions away. So the idea here is let's constrain our transferring guidance. Okay. We also have empirical zones that are species specific and based on, usually based on field-based experiments like common gardens. This is the same game that we play in terms of color matching to enhance restoration outcomes. I'm showing you two for blue bunch, wheatgrass, and taper tip onion. And by looking at the complex patterns of trait variation, if you plant seeds from multiple sources across a species environmental distribution within gardens that are also distributed across environmental gradients, you can do a bunch of fancy statistics and come up with these zones. What I'm highlighting here between these two circles on the left, you can see with blue bunch, and if you focus in on the red, right, that's generally the same geographic location as the dark brown on the right illustration. And what I want you to see is that the dark brown doesn't have the same pattern to the west as the red does to the west. And that really highlights that species have very different adaptive responses to their environments. And that's why doing these types of analyses, it continues to be important. Again, though, we have Ammonic as a constraint here in terms of, if you look at blue bunch again, if you have a restoration site in the red, the idea being it probably isn't the best idea to pull your restoration materials from Washington, from the red area in Washington. Okay. So now I'm gonna extend the idea of genetically appropriate to include genetic diversity and structure. And this is where we get some of the overlap with millions of years ago and into the past. So I'm a geneticist and a botanist and an evolutionary biologist, and I really love telling stories about the genetic variation that's within plants that you can't see, because they tell really fantastic things about history. So this was a study that we did with Guyetta grass, and what you're seeing is that the circles are where we collected molecular data. The colors represent the genetic population that plants are assigned to, and there are multiple types of analyses that go into that. But here we have 3 genetically defined populations. Plants sharing the same color are most closely related. So you can think of them as some kind of family unit almost. They share close relationships, and they share presumably ongoing gene flow. However, it's important to note, I'm not showing you anything about adaptation here. I'm showing you things about history. All of these blue plants, for example, are still distributed across diverse environmental gradients. And as a final point, this type of diversity represents a recognized form of biodiversity that takes thousands and thousands of years to develop. And here we go. So all of the species, I would wager, if not, the vast majority of species across the Intermountain West show patterns like this, and they can all be tied back to changes in distributions during the Pleistocene over the past 2.5+ million years. So during these times, as glaciations caused vast changes in our climates and plant communities, plants had to move their distributions in order to survive. So the plants we see in our landscape today are the survivors from having to move up and down elevational gradients, north and south, east and west over time. And the pattern of colors that you see on the left here is directly tied to that, especially in the West, where we have all of this topographic heterogeneity. You can imagine things being split apart by mountain ranges during certain parts of their history, whereas they would come together during other climatically different parts of their histories. Okay, and why does this matter for restoration? Again, it's a form of biodiversity. Genetic patterns may reflect taxonomic diversity. This was hypothesized in, for example, in Erigeron speciosus, where these genetically defined populations seem to line up with varietal status. This is so more distantly related plants have higher chances for outbreeding depression. This is a cool example from an orchid in Brazil, where the colors that you're seeing here are similar to the colors that I'm showing you up in the upper left. But what these authors did is they selected plants from all of these different groupings and started manually crossing them. And that's what you're seeing in the figure on the left. And what you really need to focus in on here is that the width of the bars represents how compatible the plants are. So how viable the seeds are and how vigorous they are when they germinate and establish. And what you find is that if you do crosses with plants that are more closely related, such as plants that are the same color, you have good crossing success. But as you start crossing plants from different color groups, it becomes really unpredictable. And that's a representation of outbreeding depression. And outbreeding depression isn't necessarily a bad thing, but when we want our restoration materials to work in our treatments, when we put them in, if they're cross-pollinating with pre-existing plants or naturally occurring plants, and they're not related and they have lower seed viability and the seedlings aren't vigorous, you're providing opportunities for invasive species, for example, that don't need to be there. Individuals, so I have a lot here because this is really exciting to me, but I think we're gonna have to go a little bit quicker. This bullet point really refers to the fact that you can have plants within a species that come up with different adaptive solutions to the same environmental problems. So there's a lot of adaptive diversity in plants that's really important to think to acknowledge in terms of preserving some of the natural patterns. And finally, members of the larger communities like insects may have preferences on certain plant genotypes. And you can think about this as you know, one insect might not like the taste of native plant material 1 because it's not used to it, where it does like the taste of native plant material 2 because it's more local. So all of these things are reasons to incorporate historical patterns. How do we do this? Well, we've come up with a really complex methodology, which I'm not going to tell you about, but the whole point of this is that you translate these patterns of genetic diversity into shapes that you can then use in your restoration guidance. And you also have confidence implicit within here, such that you can actually, if you find plants within one of these colored polygons, you can not only talk about what its potential identity is, but how much confidence you have. And the next step of this is combining what I'm calling a historical provenance with an adaptive provenance. And provenance, it's used in various ways in the literature, but here I'm providing a definition up top from a paper from Martin Breed and colleagues. So what I showed you is basically how you can estimate a historical provenance, and the adaptive provenance optimally would come from a field-based study like a common garden experiment. If you don't have that, you can use genetic data to estimate it as well. And there are examples in the literature where, in general, genetic data can come up with similar solutions as common gardens. They're definitely not one and the same. So from here, if you superimpose your historical provenance on top of your adaptive provenance, you can all of a sudden have seed transfer zones that include both. So you're taking into account that historical diversity as well as adaptive diversity. And this is what we've been doing for Colorado Plateau. And there's actually, I think, 8 species now available on the Western Wildland website. And the important distinction here is, the important point here is that Amarnic doesn't come into this solution anymore. By accounting for the historical provenance in this way, we don't need to think about whether or not things are in the same or different ecoregions anymore. And that is what actually ends up simplifying guidance in terms of the genetically appropriate paradigm. Again, the genetically appropriate paradigm suggests that we're using materials that are constrained by Amarnic level 3 ecoregions as a conservative way to try to protect some of these patterns. For blue bunch wheatgrass and these empirical zones here, there are 9 ecoregions that the species is distributed across. We have done a landscape genomics for blue bunch, and there's actually 5 populations. And if you lay those over the adaptive surface, you have far fewer future plant materials that you have to worry about developing. So these don't quite fit on top of one another. You can see that, but with genetic data, it's easy to collect more sites from outside of your study to extend boundaries of historical provenance. So, oops, wrong way. All right, let me see here. I have a few more slides. I think I'm just about out of time, but I'd like to at least mention them quickly. This was more put in here to talk about some of the like how we actually use all of our materials to guide our restoration treatments. So first of all, when we have to do restoration and we have to select among native plant materials that exist, right? That's what we have available for us. And if we're following genetically appropriate guidelines, we would want to use seed transfer zone concepts. You can use, there's a seed zone mapper available at the Western Wildland website, and that includes some but not all seed transfer zones. There are a lot more that are stored there that you can, if you know how to use shape files, you can download those so that you can guide your material selection and use. So, but oftentimes I think most of us know that there might be materials available, but none within a specific zone. In those instances, you can do environmental matching. So this is more of a focal point approach where you look at environmental similarity between plant materials in your restoration site without necessarily worrying about the zones. I'm showing you an example here. You could use something like the Climate Smart Restoration tool for this. I'm showing you a different tool that we've been working on here at USGS. But what you end up doing is putting, in this example, you're putting available germplasms in the context of the genetically appropriate paradigm and ranking them environmentally in terms of your restoration site. Sorry. And this can help you prioritize, like if you have the ability to purchase Delta, Bonneville, and White River, you might look for Delta first if it's available. And finally, the last part of this is, which many federal folks and researchers are currently involved in, is developing new materials to really support future restoration. What I'm showing you on the right here is basically a gap analysis where we looked at what materials are available and where the disturbances are. And so what you're seeing on the bottom here with Sphaerosia parvifolia is that there are no materials available. The red hashed is where there are disturbances. And I am just providing an example of using the genetically defined seed transfer zones. And with Colorado Plateau, we chose several seed sources that were within that zone and sent them to a grower through the BLM's IDIQ contract process to increase this seed as a native plant material that would be available for use throughout this zone 3, and especially for that vernal area that has a lot of oil and gas use. Okay, so I think I am done. Genetic data are awesome. There are lots of opportunities with them. And by including them, I think you can let people within the science and research fields worry about how to do it, but doing it can actually make things easier in certain ways and hopefully increase successful outcomes. And that's it.
Thanks, Rob. That was so neat. And Corey put the link to that website in the chat, and I'll try to get it up on the follow-up email as well. All right, thank you so much, Beth and Rob. And if you have questions for either presenter, please type them in the chat. The first question is for you, Rob. I'm not in the Great Basin, up in the wet Pacific Northwest, and effects of climate change are increasingly obvious in both extreme, outside the range of historical variability events, and rapid shift in winter precip and summer heat. Oh, sorry, let me turn my video back on. There we go. Any words of wisdom in terms of how far afield from a restoration site is it appropriate for seed provision?
Yeah, changing climates are just quite a thorn. One of the biggest challenges is that even if like climate averages are changing, you still get those extreme years that can make it really difficult to use a material that might be more suitable for a future climate. But I guess to answer your question more directly, we have other guidance, or I've looked at genetic patterns across species, and there's an equation that you can think about in terms of how far afield you can go to look for plant materials and still try your best to account for those natural historical patterns of genetic variation. And I mean, it comes like, it's something like, at 200 kilometers, you have like a 50% chance of sampling something that's very genetically distinct. If you're interested, I can provide that for you, reference for you, but yeah, I think that's.
Okay, great, thanks. Fantastic work. Do you know of any efforts to develop these kinds of tools for other states, particularly California?
So for tools, so the climate smart restoration tool, which Bryce Richardson and Francis Kilkenny and others have been working on, is West-wide, and it's really great because it includes, you can actually account for future climates as well. So so certainly that would work really well for California as well. And like, so what I showed that I'm working on, that's North America wide. So I think a lot of the tools that are available, you can really use them throughout most of the areas of interest.
Great, thanks. And is it okay if I put you in touch with the person who asked the previous question, so you can help them out with that broad scale guidance?
Yep.
Excellent. Comment very helpful as EcoSource proceeds into the production aspect of ecological restoration, that too. Yeah, any other questions? Presenters, do you have questions for each other?
I love being freed from thinking about ecoregions. I'll say that. And I feel like I have to think about that. Yeah.
So if you look at enough species, and there's been tons of work, so looking at the historical patterns of species and understanding the factors that have influenced them is called phylogeography. In the West, there's been tons and tons of examples, and by and large, they almost never follow ecoregions. And in my opinion is that we should stop talking about ecoregions as a constraint and instead be thinking about what are those adaptive surfaces like, from common gardens that in best circumstances, and then layering on top the layers of historical provenance, and it's just going to provide so much more. I mean, Beth, so you talked about these patterns over 13 million years, and thinking about what we want our species to look like into the future. It seems like maintaining those innate patterns of diversity are really important, and really having that data is the only way that we can provide that level of protection.
Yeah. And thinking about where they spent the last ice age and retreating and recolonizing, I mean, that those processes, they're hopefully going to be ongoing, and we want them to be doing those retreats and with their close relatives. Yeah.
Well, we should be going back into a glacial period now, and it feels like we are in Flagstaff at least. So we'll see what happens.
Same in Reno. Question for Beth. Our seed shattering.
Yeah.
Do you recommend harvesting the seed heads and placing into boxes on multiple dates during the seed maturation window?
Yeah, that's what Tara did. She did it in sort of an experimental way, seeing like, when do you get the most viable seeds? If you harvest too early, they're not going to ripen. If you harvest too late, you miss too many. And so there was like this Goldilocks window of like, okay, you harvest at this time, that's when you get the most seeds. And that's kind of like, it's a little bit, of a slog to do that work, but it's better to do that than like spend millions of dollars into it. You know what I mean? So we need this like very basic R&D. When do you put it in a box? You know what I mean? Yes.
Yeah, that's great. For the homeowner who just wants to improve her landscape for beneficial and wanted insects, What do you recommend as far as plant seed sources? I don't want to collect seeds, but I do want appropriate seeds and plants.
If you're lucky, you have a local native nursery or a local native plant society. If you're in California, you 100% have that. If you're in Nevada, you probably don't. I hear that. But your native plant society, they all collect seeds and give them to each other. So find your native plant society.
Yeah, because we have some of that in Nevada, right? Yeah.
You know the right people.
Beth, do you have thoughts on economic or funding innovation that may help with innovative ag production?
Yes, the USDA could easily make this a priority area for grants and research. And then, just as they did with pollinators, do you remember when the USDA decided that pollinators and pollinator habitat was a thing? Then all of a sudden that funded all this sort of R&D. So if USDA decided that native seed production was a priority, that would do it.
Cool. Any additional resources or discussion regarding using seed zones when developing new materials?
So there's internal discussions about the best way to use seed zones, and in terms of you know, the BLM and the IDIQ. I don't know that. So if you know if you're in the federal service and know some federal folks, you can start asking questions. If you're not, if you go to the seed conference or other conferences, there are workshops. I don't know that there's necessarily a guide out there that can help you like step through kind of what the decision making might be, and that would be a great thing to do for folks that are a few steps away from government.
We need to form a discussion support group.
And I think, you know, really comes down to like color matching, if you have the right zones or a provisional zone. And if you don't, you know, be familiarizing yourself with those tools and being able to figure out how to environmentally match those distances. And, you know, I'm a scientist, so this sounds easy to me, but it's not so intuitive to everyone else. And you know, Brights, for instance, this tool has a great user guide, and the tool we work on has a user guide. So I think there are resources out there. It's just, and you can contact me. I can point you towards things. I don't know that there's anything real easy though. Sorry.
I do too. Need a native seed support group.
You know.
That would be great. Okay, yeah, let's see. Yeah, all right. Thank you, Lauren, for someone asked for a link to the Native Seed Conference, and Lauren put that in there, and it looks like Corey did too. Thank you both. All right, well, that looks like the last question. Great discussion, and thank you all for your participation. We would greatly appreciate it if you would take our 3-question survey that will appear after I close the webinar. This recording will be posted on our Great Basin Fire Science YouTube channel. And Corey, I don't know if you want to drop that YouTube channel link in the chat now, but we'll also send it to you after the fact. And that'll be in the next few days. The link will be automatically sent to you through this Zoom webinar, and we'll try to also put all the resources that were in the chat in that follow-up email as well. We hope you can join us for the next webinar on seed modifications. If you have further questions regarding today's or other webinars, please email or call me anytime. Thank you all for attending this webinar, and thank you, Beth and Rob, for presenting. This is really great. All right, have a great day, everybody.
Bye. Thank you.
Bye.